Welcome to the deep dive. Today we're diving deep into sepsis. It's a global health threat I'm sure most people have heard of, but maybe don't fully get, and how it's this race against time, and researchers are always looking for better ways to detect it and, of course, combat it. And that's what today's deep dive is all about. We're looking at the science of lipopolysaccharide, LPS for short.
EPS.
Yeah, LPS. It's a key culprit in how sepsis, takes hold.
Right.
And we've got some really interesting research to unpack, specifically a 2019 paper by Coniam and their team about a potential breakthrough in, well, LPS detection.
Yeah.
So what's got you so, interested in this paper?
Well, this research really caught my eye because it tackles a super critical challenge in sepsis care, and that's speed.
Okay.
So imagine LPS as kind of like a molecular alarm bell that goes off when certain bacteria invade our bodies.
Right.
But the problem is that sometimes our body's response to that alarm, that immune response we hear about, can be even more dangerous than the infection itself. Oh, wow.
And that's where sepsis comes in.
Okay, so it's not just the bacteria, it's our body's reaction to it.
Exactly. And what's so fascinating about this, what Coniam and the team have done, is they've developed a new molecule called BPTG, and it acts like a homing beacon, lighting up in the presence of LPS.
So instead of waiting for those alarm bells to, turn into a full-blown four-alarm fire, BPTG could help us detect the threat earlier.
That's exactly it. Early detection is key with sepsis. I mean, really key, because it often can mimic other conditions.
Oh, really?
Yeah, making it really tricky to diagnose quickly.
Hmm.
And this paper, it does a good job highlighting how severe sepsis is. It affects millions of people globally every year with a tragically high mortality rate.
Wow.
A faster, more accurate diagnostic tool, game changer.
Yeah, that really puts it into perspective. It's not just about understanding a molecule. This is potentially, life-saving stuff. But how does BPTG even work? What makes it so special?
So existing methods for detecting LPS, they have limitations.
Some are really time-consuming.
Right.
Others just aren't sensitive enough. This new research introduces this concept of a fluorescent sensor, which is where BPTG comes in.
Okay.
Think of it like this.
Yeah.
Imagine you're shining a black light, on a hidden message.
Yeah.
BPTG works in a similar way, but instead of revealing a message, it lights up when it encounters LPS.
So it's like a molecular spotlight highlighting the LPS so we can see it.
You got it. Precisely. And the way it does this is, it's quite elegant, actually.
Oh.
BPTG has this very unique structure, almost sandwich-like. And when it bumps into LPS, it binds to it. And that triggers this turn-on mechanism, causing it to fluoresce.
A turn-on mechanism, huh?
Yeah.
Tell me more about that.
So this turn-on feature basically means that BPTG only fluoresces when it's actually bound to LPS.
Oh, okay.
That means it significantly reduces the chance of a false positive.
That's huge.
Right. It's like having a smoke detector that only goes off when there's an actual fire.
Right.
Not just, burnt toast.
Yeah.
And that high level of accuracy, that's crucial for something as time-sensitive as sepsis diagnosis.
That's a fantastic analogy.
Yeah.
But how does BPTG, how does it know it's, found LPS and not some other random molecule in our system?
That is another remarkable thing about this research. BPTG shows incredible selectivity for LPS.
Oh, wow.
Meaning it can pick it out from a huge crowd of other molecules. Think of it like finding a specific grain of sand on a beach.
Right.
But in this case, the grain of sand is LPS, and the beach is our incredibly complex biological system.
Wow, that's some serious molecular detective work.
Yeah.
I remember reading something about heparin in the study. Why is it important that BPTG can, differentiate between LPS and heparin?
Great question. So heparin is a medication used to prevent blood clots, but its structure can sometimes be mistaken for LPS by other detection methods.
Oh.
BPTG, however, can easily tell them apart.
Wow.
And that's essential for accurate diagnosis and, avoiding unnecessary treatment.
So it's like BPTG has X-ray vision seeing past the surface to identify its target. With incredible precision. This all sounds, promising in the lab. But the research didn't stop there, did it?
You're right on it. The researchers took this a step further and actually used BPTG to visualize, get this, live Vibrio cholerae bacteria.
Whoa.
The major culprit behind cholera.
So instead of just knowing LPS is, present, they could actually see it.
Yeah.
That's pretty incredible.
Right.
Why is being able to visualize it, LPS in action. Why is that so important?
Well, this visualization capability, it opens up some really exciting possibilities, especially for understanding how infections actually take hold.
All right.
Researchers could track the movement and behavior of bacteria in, well, real time, almost like watching this, microscopic battle unfold.
Oh, wow. That's amazing. So it's like having a front row seat
Yeah.
to observe how these bacteria operate, how they kind of interact with our cells, and then how we can stop them.
Exactly. And just imagine what this could mean for diagnostics. Being able to just quickly confirm the presence of, these bacteria, say in a blood sample.
Right.
That could be revolutionary for a condition like sepsis where every minute counts.
Yeah, absolutely. It's like upgrading from a blurry map to a high definition, real-time GPS system when you're trying to find something.
Yeah.
This BPTG molecule sounding more and more like a game changer.
It's really interesting stuff.
But I'm curious, what are the next steps here? How do we go from this promising molecule in the lab to something that can actually help people?
Yeah, you've hit the nail on the head. It's exciting, to consider the potential here, but it's really important to remember that this research, it's still early stages.
So what needs to happen next? What are the hurdles that researchers need to clear before we can get BPTG, from the lab bench to the bedside?
Well, one crucial step is to see if BPTG, it holds up in more realistic biological environments.
Well, right.
I mean, the lab is one thing, but the human body, that's incredibly complex.
Yeah.
Researchers are going to need to test BPTG's effectiveness in blood samples from septic patients.
Okay.
And in animal models of sepsis, see if it actually performs as well in those, real-world scenarios.
Right. It's like taking a promising athlete from practice to the big leagues, right?
Exactly.
You got to see how they handle the pressure.
Precisely. And another area of focus is going to be optimization.
Right.
Fine-tuning BPTG's structure
Okay.
to enhance its sensitivity, maybe stability, or even its ability to penetrate those bacterial cell walls we talked about.
Right.
It's kind of like, imagine a mechanic tweaking a race car's engine just to get that absolute best performance.
Yeah.
That's what researchers will be doing with BPTG, making it even faster, stronger, more adaptable to those challenges of the human body.
So it's about pushing BPTG to its limits to see what it's really capable of. And, we've mainly talked about BPTG as a diagnostic tool, but could it have, therapeutic applications? Could it be more than just, a detector?
Now, it's a fascinating question.
Yeah.
It really speaks to, the ingenuity of this research. While the focus has been on detection, it's conceivable that BPTG could be modified to neutralize LPS directly, or maybe interfere with its ability to trigger that, dangerous immune response we've talked about. It'd be like going from simply sounding the alarm to actually disarming the threat.
That would be remarkable, transforming BPTG from a molecular spotlight into a molecular shield.
Exactly. Of course, that kind of application is, further down the road.
Sure.
But it just highlights the really exciting possibilities that this research opens up,
Absolutely. So as we wrap up this deep dive, what's the key takeaway you want to leave our listeners with?
Well, I think the biggest message here is one of hope. Okay. Sepsis is, it's a formidable opponent.
Yeah.
But research like this, it shows that we're making strides towards faster, more effective diagnosis and treatment.
Right.
It's a real testament to the power of scientific curiosity, innovation, and that relentless pursuit of solutions to some of, humanity's biggest health challenges.
Absolutely. Well, this deep dive has been, quite a journey from, the intricate world of molecules to the front lines of fighting this global health threat.
That's right.
We've seen how a deeper understanding of LPS could really hold the key to earlier diagnosis, more targeted treatment, and potentially, even preventing sepsis altogether.
Absolutely.
Well, thanks for joining us on this exploration. Until next time, keep those brains curious.